Alright guys, welcome back. So let's continue on with this series. As usual, you know I'm doing the new series of videos, different from how the previous ones that got copyrighted were. So now I'm just talking off of things that I teach. I have my own First Aid 2021 open on another monitor below. I'll be referencing to you the pages that I'm using from First Aid 2021, and if it differs from my annotated First Aid, which I have extra pages in, then I'll definitely make sure to clear that up as best as I can. And of course, as always, feel free to shoot me an email if you want to sign up for my Step 1 course, my Step 2 CK course, or want to buy any of my annotated products for perfect studying for your step prep. Now, right here, last time we left off at the conversation where we wrapped up talking about DNA repair. We went back and revised a bunch of stuff from Fanconi anemia to all the different kinds of repair mechanisms and where they play a critical role. Now, today we're moving on to the topic of organization of a eukaryotic gene, which will then follow suit into the tRNA and all the other things that happen to a gene from transcription down to translation into a full-fledged functional or non-functional protein, depending on whatever happened. And of course, as we keep going on, I'll be talking about how questions will present about these topics. So right here, we're going to be looking on page 41 of First Aid 2021. So let me put that into the note. Right, and we're looking at a eukaryotic gene. So it looks a lot like what I've written right here, right? So you can see they've labeled for you the different portions of this gene, right? So you have your, let me grab my pencil. You have in the beginning, right after the 5' region area, anywhere here, you can have enhancers and silencers, right? Enhancers and silencers are filling up that area. These enhancers can also be found in another place, anywhere throughout this, right? So not in the center, but like anywhere before this entire start and end area. So the end, let me put in the end in here too. So ending will basically be sometime right after that last exon, right? So that would be where the ending would be, and then you add a poly A tail as a signal, which often does get expressed actually. So you know what? Let's move this over. So that's a pretty good point to keep in mind. The poly A tail is also actually going to be oftentimes part of your exon. You can actually see that in that row where they have this line written. They wrote the portions that are considered exons and introns in the book, and the last exon actually encompasses your poly A tail, right? So the poly A tail often is expressed, which actually gives not exactly a part to the final protein, but it will create what you will have in the end as your final mRNA. It's going to be part of your final mRNA, and then that's going to go into being transcribed, but the poly A tail portion isn't really going to yield a significant part of the final product, the protein that you're going to have at the end, right? So you can have enhancers here, right, anywhere before your start, and then you can also have enhancers anywhere after the end, right, as well as silencers, both things. So in the book they've only written the silencer, but enhancers and silencers can actually be on either end, right? So they can be anywhere downstream or upstream from the start and end portions, right? So you have your start, you have your end right here. Enhancers and silencers can be anywhere. Now what they do is they further, as the word would say, they further enhance or suppress, silence, the actual transcription of this area, right? So whether or not this area will even be transcribed by RNA polymerase and stuff like that depends on whether or not the enhancers are really going to be making that go faster, right? So if an enhancer is not working, transcription will occur, it just won't occur as quickly, right? Whereas if an enhancer is working, then the transcription may be amplified by 3 to 4 times, maybe even 10 times more. So that's one important thing. Then you have these guys, right? So you have the cat box and you have the TATA box. So what are they all about? Well, there's a question that goes on talking about how a patient with chronic anemia comes to the clinic, they need chronic blood transfusions because of that anemia, and then the question all of a sudden shifts gears from being all about this anemia, a hematology chapter topic, to suddenly talking about sequencing of genes, right? And they really focus on the beta globin gene. So imagine in a world where you get a question, imagine on your step you get a question about anemia, right, that then translates into a question about the beta globin gene. you already know this is going to be a question about beta thalassemia. Furthermore, beta thalassemia, we saw earlier, a couple pages above, let me see where, oh, right before, the page right before where they talk about splice site mutations, we saw that the splice site mutation is a mutation where you have a retained intron that did not get spliced out, right? Introns are intervening sequences that need to be removed. Exons are expressed. So if you didn't remove certain introns, then that can be one of the rare causes of some types of beta thalassemia. So that's the first thought that should come to your mind when a question is starting off with anemia and then beta, and then they go into talking about genes, right? Because that's what the question is doing. They suddenly shift gears. They're talking about sequencing of genes. Then they talk about how they're pointing at the same image we have up here, the same little 5' to 3' thing we have here, and they're pointing at something which is actually the data box, right? They're pointing at the data box, and then they ask you that little pointed section, what is it responsible for? Well, people fell for both answer choices. There were two answer choices, either initiation or enhancer. The other answer choices didn't make any sense. So is the data box an initiator or an enhancer? Well, we just finished talking about where the enhancers are, right? The enhancers are either upstream, very close to the 5' area, or all the way downstream past after the last exon. So the cat and data boxes are actually initiators. Those are the guys that initiate and start transcription. So it actually goes in that order. An enhancer will upregulate the cat box, whereas a silencer would stop it. A silencer would slow down the cat box and the data box, right? And then the cat and data box together will upregulate and start, right? Initiate the actual transcription that's occurring, right? So the enhancer and the initiators are two different guys that will always be tested in summation together. You know, you're going to have both answer choices there, and the question is often going to be a big paragraph, which is really going to try to confuse you into thinking about all kinds of things, throw you off guard and whatnot, and then just try to catch you. But you have to be ready with that, right? So the enhancer is something that can be on either side, and so can the silencer, and those are guys that just enhance or silent, but they don't start anything. The cat and data boxes are the ones that actually start stuff, right? So those are the guys that are going to be critical in actually initiating transcription, which will then begin. So now you can see another critical thing to point or pay attention to in this this DNA, by the way. First of all, keep in mind this is what we're looking at right here is your DNA, right? It is your DNA that would have all of these things. The DNA is the one that's going to choose whether or not transcription should occur. So that's what we're enhancing. We're enhancing or silencing transcription, or we're initiating from the cat and data boxes transcription. So this is DNA. So on your DNA, the other thing you're seeing are these segments right here that had the GTAG, right? These segments that have the GTAG, those are introns, right? And the GT and AG are critical because you can always get questions where they'll tell you, you know, a long strip of DNA sequence, just like this, but like maybe 50 lines of it. And they do this oftentimes where they'll keep the exons capitalized and all the intron stuff lowercase letters, and then it'll be all like GT, GC, all that stuff, right, nonstop for maybe 50 lines. And then they'll show you that in one random place, in one of the introns, maybe this A got deleted and became something else, got replaced with maybe a C. So now your guys that are supposed to work on splicing and removing introns and all of that will no longer be able to recognize that intron. So what will occur? A splice site mutation, right? And that splice site mutation will be something that can lead to possibly beta thalassemia, can also lead to all kinds of other things like epilepsy and whatnot, right? So we've seen some of that when we looked at splice site mutations earlier. So that's a critical thing to watch out for. It's really important to remember the GTAG. I just remember that as the G tag, right? Which is very easy to do that. Now, the G tag is something you see on DNA. When you move over to RNA, all the T's are going to become U's. So that's something you will always have to naturally remember. So as long as you remember G tag, depending on whether you're talking about DNA or RNA, on RNA it will be GUAG, right? Which is still G tag in a sense. It's just you know there's no T's on RNA. So just keep that same memory going, and then you should be able to grasp all of that down just fine. So what's supposed to happen from DNA to RNA is you end up canceling out all of that stuff, right? So if I grab this and I copy and paste it again, right, let me just make sure my face isn't blocking it. All right, great. So now, what are we going to do? Well, on our RNA, there's a couple things we're not going to have. We're not going to have any of this, right? We're not going to have any of the enhancers, the initiators, silencers. We're not going to have any of that stuff on our initial transcription. Transcription just occurred. Our initial transcription is not going to have any of that. And it's also not going to have this portion over here. So really, all we did was we grabbed everything that was from start to end, right? Which include all our exons, all our introns, the G tags, and then the poly A tail. This thing is your pre-mRNA. Right, so now we have our pre-mRNA, which is basically premature mRNA, if you want to call it that. You know, if you want to think of it like that. You could think of it as premature mRNA. It's just not perfectly ready yet because it still has these intervening introns. And also, one more thing we need to edit now, since this is RNA, the T's are now going to be U. Right? So all the T's, every single T anywhere became a U. So now you have your pre-mRNA. Now what's going to happen with this? Well, we can copy paste this one more time to see what would happen when this becomes mature RNA. And the only thing that has to occur successfully to make a mature RNA is just getting rid of the introns. Right? We're just going to get rid of all of our introns right here. And look at this. Now we have this tiny thing that is our mature RNA. It has absolutely nothing to it. There's nothing on it. The only thing it has are the exons that are going to be expressed and the poly A tail. Remember that the poly A tail stays on this because it has one very functional reason. The functional reason this thing even exists in the first place is for stability purposes, right? The poly A tail will keep this entire thing stable, and that's all it really does. It doesn't have any other function as far as transcription and translation are really involved. So if you ever get a question asking you what the hell this is for, it's really just for stability, you know, or pick any other answer choice that goes around that nature of stability. That's all you're really looking for. And this is it. This is what's going to now be sent to be worked on by translation services, which are going to occur in your ribosomes, right? So now after this, I want to make sure I make a good point talking about some terminology things that you can get tested on. So if we look at this image up here, let me make it a little bigger. So this image here is showing you your DNA on the top, double-stranded, right? Going from 5' to 3', that's your sense strand. 3' to 5' on the opposite side is the antisense strand. That's your double-strand DNA. Now, transcription, whenever you do transcription of a certain DNA strand, that DNA strand that you're transcribing is called the antisense strand. Right? Transcription is going to occur of the antisense strand. The reason you do that is because when you actually transcribe it, look at what occurs. You see the 5' to 3' sense strand and the new mRNA that you made, which is basically your premature mRNA, it's the same as your sense strand. Everything is exactly the same as the sense strand except the T's became U's. So I always like to say DNA makes sense, right? If you say DNA makes sense, that means that DNA is going to become the mRNA that mimics the sense strand. Your mRNA is going to mimic the sense strand. And the only way you can mimic the sense strand is if you use the antisense strand as a template for transcription. Right? Because you have to copy the other thing to reinvent the other guy. Remember, coming back to the DNA, imagine that something occurred where maybe you had a mutation in your sense strand and these guys were lost. Well, how would you fix that? Well, to reinvent the sense strand, you would just look at the antisense strand and then fill in the gaps in the sense strand based off of that. So you would see the A here and you would put a T there. You would see the C there and you would put the G there, and then there you go, you fix your sense strand. So anytime you're trying to copy and recreate the sense strand, to do so, you have to use the antisense strand as a template. So transcription occurs of the antisense strand, and the mRNA you make, it looks just like the sense strand. This is something you have to get comfortable with talking about. Just make sure the terminology, the vocabulary here isn't very confusing to you. You should be very confident in talking about it. DNA makes sense. When you transcribe DNA, the mRNA that you make is going to look like the sense strand because transcription occurred using the antisense strand. Boom. That's it. Very simple. And then this is where we basically stop, where after doing that, you remove all your introns and you get your mature RNA. and then you're going to go into translation. So I put a couple notes about that just to make that very clear. The next half of this page talks about regulation of gene expression and then RNA processing, which is all the things that are occurring on this page and the stuff we just did, right? So this is what we're looking at right now. So regulation of gene expression, there's three things here. There's a promoter, an enhancer, and a silencer. We talked about the enhancer and silencers. We also talked about the initiators, right, which was these guys. The CAT and TATA are initiators, right? These are initiation. All right. So then where is this topic of promoter coming from? So they tell you that promoters are sites where RNA polymerase II and multiple other transcription factors bind to DNA upstream from the gene locus, which differ between eukaryotes and prokaryotes. So these are upstream from the AT-rich TATA and CAT boxes. So it turns out that your promoters would also be in a similar area, right? In a similar area where enhancers and silencers are upstream from the CAT and TATA boxes, which are very TA-rich, right? As you can see, this is a pretty common theme to have a lot of ATs on both ends. Once again, generally speaking, for just stability purposes. That's all they really do. So your promoter is just that site where you can bind there, get a bunch of transcription factors there, and then basically promote gene transcription. Promoter mutation commonly results in a dramatic decrease in level of gene transcription. All right. So now, how is that different from enhancers? Well, enhancers are a DNA locus where regulatory proteins like activators bind and increase expression of a gene on the same chromosome. So generally speaking, they seem to be very similar. The only difference is what's actually binding, right? So it's a very technical difference. So just to repeat that, enhancers are where regulatory proteins, which are also being called activators, those are what's binding in the enhancers. In the promoters, you actually have transcription factors binding, right? So the actual thing that's binding to the region is what makes it different, right? So enhancers, we have activators binding, right? Activators are going to bind the enhancers to upregulate that. But for the initiation areas, we're going to have transcription factors that are binding. All right? So those transcription factors are going to be all kinds of things, very little tiny things that come together and then make a little complex, and then that attaches to that area before the CAT and TATA boxes, and boom, you have your promoter doing its job. All right? Silencers, on the other hand, are going to be a DNA locus where regulatory proteins known as repressors are going to bind, decreasing expression of a gene on the same chromosome. Enhancers and silencers may be located close to, far from, or even within, inside an intron whose expression they regulate, right? So, turns out that enhancers and silencers could be inside these introns, right? Way big in the beginning, many years ago, when we were still really behind on a lot of this research, and step one used to be a lot easier. Introns were actually thought to be completely garbage DNA, but that turned out to be false because introns actually carry a lot of useful information. They just don't make protein. That's it. They just don't actually go into the mature RNA, which will ultimately be translated into a protein. But introns are still very much significant, very much significant. So now if you ever get a question where you have answer choices that are enhancers and promoters and initiators, right? So now we got three guys we got to talk about: an enhancer, a promoter, and an initiator. The initiator is very unique. That's the guy that started your actual transcription. It's a part of the DNA that is either the CAT or the TATA box. That's it. Those two guys are actual initiators. Promoters and enhancers come before that. So promoters would also be somewhere right here, in this area, before the CAT and TATA box, right? And in that promoter, you would have transcription factors that are binding, right? Transcription factors that are binding there. So now, if you have to pick between one of the three as an answer choice, then it really depends on what they've given you. If they tell you that it's transcription factors that are binding, well, transcription factors are going to be related to the promoter. So you're done there. You pick, it's a promoter sequence. Whatever part they're pointing at must be a promoter sequence, and you're done. If it's a bunch of activator proteins that are binding there, regulatory activator proteins, then it's an enhancer. Regulatory silencer repressor proteins, then it's a silencer. And if they tell you that there's a certain thing in one of the introns that is somehow influencing DNA transcription, then whatever's in the intron must be a silencer or enhancer. Really depends on what the influence is. Is the thing inside the intron causing more DNA transcription? Then it's an enhancer. Or if the thing inside the intron is causing decreased DNA transcription, then it's a silencer, right? So that's the main concept of those three terms. They are different from each other. You're going to have to know the key little technical differences between these things, because that's what you're going to be given to be able to pick one over the other in your answer choices. And then the last thing they talk about on this page is RNA processing, right? So now RNA processing is your next thing. What is this? Number 10. So 11, RNA processing. Okay. So this is just to keep in notes to see what we're doing, right? So I can know where we left off and for you guys to be able to follow along. This is still on the same page. They tell you that the initial transcript is called the heterogeneous nuclear RNA, which is basically this very first thing. The pre-mRNA that you make is a heterogeneous nuclear RNA. That thing is then going to be modified and becomes mature RNA. And this modification that occurs is going to occur inside the nucleus, right? So hnRNA becomes mRNA in nucleus. How does it occur? Well, there's three steps that have to occur. Three steps that we did here as we were deleting things. First, you're going to have a capping of the 5' end, right? Capped 5' end with your 7-methylguanosine. Right? And then on the other end, you have to do, as we saw, the poly-A tail. So you're going to have polyadenylation of the 3' end with almost up to 200 plus A's. Okay, and then the third step, the third step is going to be removing all of those GUAG introns, right? Splicing out introns. Excuse me. Splicing out introns. Okay, so that now will finally give you a capped, tailed, and spliced mRNA. So one of the most important things to know here, the most significant thing in this entire thing, is the fact that this occurred in the nucleus. So in the beginning, we talked about exceptions to DNA, right? And some of those exceptions to the general scope of DNA were the exceptions that occur in mitochondrial DNA. So if you ever find mRNA that is not capped, tailed, and spliced, but is floating around in your cytoplasm, that mRNA must have come from the mitochondria, which is often denoted as mtDNA, mitochondrial DNA. Mitochondrial DNA will not have the capped, tailed, and spliced activity occurring, because the only way you can even do this RNA processing is within the nucleus, and mitochondria are outside your nucleus, right? They're not inside the nucleus, so they can't have their stuff processed. mRNA is supposed to be transported out of the nucleus into the cytosol, and then outside, once all of this comes out, it can be stored inside something called the P-body. There's a bunch of P-bodies out there which are processing, cytoplasmic processing bodies, and inside of that there's a bunch of enzymes that are going to keep that mRNA stable and just keep it there. Storage. It's really just a storage. It could also degrade it if that stored RNA is not being used at all. You're not translating it, then it could be degraded and gotten rid of. But these P-bodies are significant for that quality control aspect. And you could get a very specific question on that, which again should be pretty easy because it's such a specific question. So you'll just pick the cytoplasmic processing body and you'll be done. Now, there is one thing that was tested pretty frequently. There was a question that pretty much only 1/3 of the students got right, where they talked about how they took some DNA, right, and then they went ahead and mixed it all up and put it together with a bunch of thymidine residues, right? So you take a bunch of, wait, let me just make sure. Was it DNA or... Yeah, so they actually took a bunch of nucleic acid from a cell they ripped apart, right? So you get this experimental question where they took a cell, they just ripped it apart, ripped up the nucleus too, and then took all the nucleic acid things out and put it all together into a little collection unit where they also added a bunch of deoxythymidine residues. So thymidine residues are a bunch of T's, basically, just a bunch of T's. And then they shook it all up and they mixed it all up, and then they ask you what is the T going to bind to the strongest. So this tail of T's. So imagine I take a cell and I find a bunch of this first thing, a bunch of the second thing, and a bunch of this third thing. Right? A lot of it. And I take all of that and I put it into a machine where I'm going to mix it up with a bunch of T tails I made. Right? A tail of T's. That's it. What is it going to bind to? Right? After all the washing and all the mixing, what is it going to bind to? Well, it makes sense that all of this is going to go ahead and bind to this, which has a bunch of A's. You know, there's going to be a tiny kink where there's a T in both sides and that part won't connect, but all the A's will bind to all the T's. This should have been a very easy question, but people got very confused very quickly, right? Very confused very quickly because of the answer choices and the way everything was worded and whatnot. But the correct answer was basically that it would bind to mature mRNA. That would be the #1 site that this would bind to because it's just easy to. It's small, it's stable, right? Remember that a very long thing is not very stable. If you have like, one of the simplest ways to test this out is if you were to take a bunch of pencils, unsharpened pencils, and try to hold them all together to each other. I wish I had something that I could show it to you with, but if you were to basically take, like, let's imagine all three of these were pencils, we would connect all three of them right here, right, from like this, like that, and I would try to hold it. You see how the middle one fell apart? I would try to hold it together as I go further out, and keep adding more and more. If you were to actually do that, the more things you would add to the middle, and the longer this sequence got, the more unstable the middle would become, right? And it would break. But after removing all the introns and removing everything else, you see how this became really small? This is very stable. So in the mixing process of mixing a bunch of T's to all of these guys, this T stuff will best bind to here. It can, and these are some of the other answer choices, it can bind to the TATA box and to the CAP box as well. However, it's far less likely, right? Most people chose the correct answer, mature mRNA. A lot of people chose promoter regions of DNA or the TATA box or other stuff like that. Those regions would not be so significant because they're just not as openly available, right? You see how it's in the middle? It's in the middle, not at the end. If it's at the end, it's very easy to connect to something at the end. Connecting to something in the middle, remember there's a bunch of electrical charges coming off of all of these regions, right? All of these things, each of these nucleotides have their own little electrical charge to it, their own preferences of what they want to connect to. So this polythymidine residue was actually supposed to bind best to mature RNA. And what this question was testing you on is an actual method that's used for capturing mature RNA in various cells to then see what is this cell actually translating, right? In a very experimental setting, if you had a cell and you wanted to see what kind of protein is this stupid cell making, because maybe it's a cell from a diseased person or it's a cell from someone who has cancer. Cancer actually makes a lot of proteins as well that helps them sustain and live, right? Cancer cells don't die. So to find that mRNA, all you have to do is shred up the cell and then mix that cell's nucleic acid contents with deoxythymidine residues, and it'll connect to all the mRNAs. It'll connect to some other things too, but the majority of what you'll catch will be the mature RNAs. All right. So that's it for this session, and then in the next session we'll move on into talking about the full knowledge of tRNA and how all of those guys work.